Building off primary, secondary, and tertiary structure of proteins, we find out that the functional form, so the functionality or the activity of many proteins is not that of a single polypeptide chain, so just one peptide, but it's actually an aggregate or a conglomeration of several globular peptides, and we call that quaternary structure. So when we have two or more proteins that have tertiary structure and we conglomerate them or we aggregate them together, we get what's called quaternary structure. So quaternary structure is the arrangement of subunits or peptides that form a larger protein, and the subunit is a term you have to know. So the subunit refers to a polypeptide chain having the primary, secondary, and tertiary structural features that are part of a larger protein. So when we take two or more peptide chains and we again aggregate them together to make a quaternary structure, each one of those peptides we call it a subunit. A quaternary structure is going to be maintained by most of the same forces which are active in maintaining tertiary structure. So hydrophobic interactions, hydrogen bonding, and salt bridges. This slide simply summarizes primary, secondary, tertiary, and quaternary structure. So primary structure refers to simply the amide bond. So here's an amide bond, and when we have enough amino acids put together, we can get secondary structure. Now what's shown here is an alpha helix, but we also have beta pleated sheets are the other type of secondary structure. So we'll put that in here. And then of course, once we get that long protein chain, what can happen is that it can fold in on itself, and those R groups are going to interact with each other via hydrogen bonds or salt bridges or disulfide bridges, etc. So it's going to fold in and form tertiary structure. Then if more than one peptide or protein with tertiary structure, if they aggregate together as subunits, we get what's called quaternary structure. So you can see here that in this quaternary structure that's shown here, we have kind of one here, and then we have another one back here. There's one down here, and there's one over here as well. So this one, not only does it have quaternary structure, but there's actually four of them bound together, or four of these proteins that have tertiary structure aggregated together. Now, in some cases, quaternary structure of a functional protein involves binding to a non-protein group. And this additional group, we call that a prosthetic group. For example, many receptor proteins on cell surfaces are glycoproteins. These are proteins that have sugar molecules covalently attached to them. Each of the subunits of hemoglobin, for example, is bound to an iron-containing heme group. And that's what's shown here, is that this is just a specific example chosen for this slide, is that what's in here that kind of looks like a flag or something, this is supposed to represent the heme group. And we'll take a look at hemoglobin later on. This slide just shows some proteins that require prosthetic groups for their functionality. So for example, glycoproteins, their prosthetic groups are going to be carbohydrates. And the other example that we're going to talk about will be hemoglobin, which will require what we call heme as the prosthetic group. And we're going to take a look at the structure of heme later on. A heme group is nothing more than a big unsaturated organic cyclic amine with an iron ion coordinated in it. And in the case of hemoglobin, the prosthetic group actually determines the function of the protein.